So I'm a biologist. I think it's pretty clear from the pictures. So I will put a lot of nice pictures so that to put a biology interlude. And so I'm really grateful to be here. So it's really nice to see all the work here and some of the models. And it's really, for me, inspiring to try to understand them. So it's really hard, but I'll try my best. But also it's very inspiring to have new ideas of how we can test theory in some of the models we are working on. And so I think, unfortunately, I think I will use some of the models which have been done. We have extended a little bit without much demonstrations and proof in a mathematical sense. But I try also maybe to disprove them by showing that the real data are more complicated, and maybe we try to improve that in some sense. So first, I will acknowledge the people have been working on this problem since several years. So first, the LMU, so Wolfgang Stephan and Daniel Sivkovic, who have done a lot of the work on some of the modeling and also to work on the seed bank problem. And I think a recent collaborator, Johannes Müller, who's a professor for mathematics in the University in Munich, who I will show some of the models we have done at the end, who's really a very good collaborator now, and I think we try to make some progress in the direction of understanding the data. So what are seed banks? What I want to talk about is a very strategy which is very common to many species, and it has been really well worked out in ecology for many, many years. And there are few theoretical models. So these are theoretical models of ESS, typically that people have done in the early days in the 60s, and later on there's been a revival a bit recently about that topic. And the idea is very simple, is that if the environment is stochastically variable, so if a species cannot predict how the environment will vary over time, or for example there is competition which can generate this temporal variability, then the species may evolve, or individuals may evolve a bettaging strategy so that they will leave some individuals, for example, in the soil for a long period of time, and this individual will come up later again when condition might be more favorable. And so this bettaging strategy is so-called generally germ banking, and this you can find it for many species. So especially in bacteria, it's very common. So these are different species of fungi or some bacteria which are doing this strategy, so the bacteria can switch to a state of dormancy. And I think Jochen in the next talk will talk more about that, so I will not say more. But there are also many crustaceans. So this is a little Daphnia, which you find in many ponds around Europe and North America, for example, which does this little also sleeping stage, which fall into the sediment. And then when the sediment are moved again, the individual can pop up again in the population. What I'm interested in more is what's happening in plants. And the plant have the same, of course, so we will call it seed banks, because the seed can fall on the soil and then get buried over the years, or sometimes get also unburied and moved up again by movement in the soil by many animals and so on. And of course, they can be stored in the soil for different lengths of time that we will define later. So it's a very important mechanism also for conservation biology, because for many endangered species, if you have these seed banks, that means you also decrease the likelihood of extinction of some of the populations or species potentially. So I think what is pretty clear, I think, is that the seed bank will promote the storage of genetic diversity in the soil. So the fact that you have plant seeds which fall in the soil and can remain there for a certain period of time, that means that these lineages can remain dormant and escape what is happening in the above ground population for some period of time. Right? So that means we have a time lag between what is happening above ground, so in above ground population, for example, selection or drift, and what is happening to the lineages which are in the soil. And there have been a couple of interesting aspects of that. One of them has been kind of a verbal argument early on that selection might be slower. Though I think if you want to do a model about that, and Jochen might talk about this, there is no real proof that this is really true and what are the timescales involved. I try to allude that at the end. Michael Cirelli and Bob Chensker have been very influential in looking at balancing selection. So this is a very nice species of flower that is found in sand desert in California. And there they have done a lot of experiment to show that, for example, these two morphs, so these two colors of plants, are maintained due to the seed bank over time because there is alternating conditions for each of the morph to go out of the seed bank. But if there would not be any seed bank, you would not maintain both types over time. And what I've been interested in early on is to also look at coevolutionary dynamics, so between plant and parasites, and we use dynamical system to show that this time lag can stabilize the dynamical system. So I do not pretend that we have a proof, far from that, in this model. So this is the general effect I want to discuss today, and of course, to start looking at it, I got interested by looking at these two species of wild tomatoes. So this is how they look like. So they have really nice yellow flowers and some more or less small leaves, depending on the species. And since I worked with Wolfgang Stephan in the LMU, we started working on these two species, which are found in southern Peru and northern Chile. And especially I will talk mainly today about Solanum chilense, which is found in Chile, as the name says. And what is amazing is that you find the species, you can see some of the pictures here. These species are found, for example, in the middle of the Atacama Desert, which is one of the driest places on Earth. So they are really resistant to drought. They can really survive a long period of time without water. And what was the main observation after several months we did is when we look, when we know from the ecology data, is that we know that these populations are in general pretty small. So if you look what I will call the census size, so that's the size of the plant above ground that you can see when you go in the field, and these sizes are usually pretty small. So pretty small means 10 to hundreds of individuals. And on the other hand, when we look at sequences data that we could gather at that time, so limited by Sanger sequencing, but now we have more, and we can confirm this observation that you find a very, very large amount of genetic diversity. So if we would extrapolate the effective population size, whatever this means for each of you, but let's say a population size expected, we would expect to have on the order of 1 million individuals in the whole species, and for each population, at least 100,000 also. So for each population, you find a very large diversity, even though above ground you don't see that. So I think the answer, I guess the title told you, what we wanted to know is seed bank explaining this effect of storing genetic diversity? Can we infer something about the seed bank from genetic data? So that was my main aim. And the idea is to use existing model of coalescence to try to estimate some parameters of this interesting ecological phenomenon. So what I will present now in the rest of the talk is, first I will present you some results about what inference we did and how we use sequence data to do that, and then some problems which appeared, of course. Then some really recent data set that we have where we look within this species and look if seed banks could be different between the populations. And I think at the end some work in progress and very open questions, and I would be happy, of course, for any comments or suggestions. So the first things we wanted to do is to do some inference of the seed bank parameters. So how do we do that? We use a very nice and elegant model from Kai, Krone, and Lascu from 2001. Which I think for a long time was the only model existing about seed bank until Jochen came and also did some new modeling. So what the model does is very simple. It's a version of the Kingman for the Wright-Fisher model, and you build a backward coalescent, but instead of having only lineage jumping from one generation to the previous one, then you can jump to generations which are further back in time. So each here, what I will call a cell, or what they call a cell in that model, is basically a generation. So this is generation time 0 when we can sample our lineages. So these are plants that I see above ground I can sample. And if I don't have any information about what is in the seed bank, this is empty. So they are just lineages, but I don't know them, I don't sample them. And then I can, each of this lineage can go back in time to different past generation. Okay, so by definition, each of this generation, at the moment we work in constant population size, will have a size n census, this NCS. Okay, so what the mechanism is that you can move with some probability function, so this bi, so this b1, b2, and so on, will tell you a probability, give a probability that you move this lineage back to the previous generation. So b1 means this plant, that we observe comes from a plant which has produced seeds at that generation before. Right? Then if we do B2, it means 2 generations ago and so on. Okay? So if we do this process over a so-called M window, so the M window means this is the maximum amount of time that seeds can spend in a seed bank. If you look forward in time, backward means how far the ancestor of that given seeds was in maximum amount of time in the past. Okay, so this, I will define this M also later. So we can do that for each of the plants in cell 0, exactly like in a usual coalescent, and then we move all the lineages backward. And of course, if some individuals can coalesce, have the same common ancestor at some point in the past. So the assumption is that only coalescent event can happen on the plant above ground. Okay, so only plants which are above ground can be ancestor of 2 lineages. And we will also assume that only this plant above ground can have mutation. Okay, for simplicity, I'll come back to that later. So at any moment, there are lineages in this M window, okay, and then this R lineages at some point collapses to 1, which is the most recent common ancestor of the population. And then when we have done this for one cell, we shift everything to the left, and then do that again and again until all the lineages have found a common ancestor. Okay, so I just copied from the paper itself the description. So if you want to look at the model and the demonstration of the proof of the convergence to the Kingman and coalescent, redirect you to that paper. But what is interesting is that in this limit, of course, n tend to infinity, the process is a Kingman and coalescent, but run on a slower time scale. Okay, and what is the time scale? The time scale is here denoted by this beta square, and this is the formula of beta. So you can see this is the sum of the BI, so from 1 to m, that's the maximum amount of time you can spend in a seed bank. So sum of the BI is 1 by definition, because every lineage must have come from a seed which has germinated, if we observe it, and divided by here the sum of the IBI, and this is basically the average time that seeds can spend in a seed bank. So it's a very elegant model and very elegant results, which sounds a bit intuitive, but it's nice. And the main idea is that this beta is a composite parameter, so seed bank parameter, which is a function of this BI distribution and M. Right? So this is important because this 2 parameter matters for defining the beta. Now what I will assume in the rest of the talk when we use that model is that we assume that the M is small and finite on the average of, let's say, less than 1,000 if you want a number, usually much smaller even for plant species. And I think I'll leave Jochen to discuss this question for the next talk where he will show what happens in the limit where M tends to very large number. Okay, so what we wanted to do is to use this model to define a bit for our purpose and to start doing inference. So what we did is to define a very basic geometric distribution of the germination rate, so to define the BIs to have it simpler. So that means we assume one probability B of germination. So we simplify in a way the BI distribution. What are the assumptions of the model is that it's clear that the rate of coalescence now is rescaled by beta square, or in fact by 1 over beta square. So the tree is getting much longer because the probability to coalesce basically depends on 2 lineages to go in the same time in the same ancestor. And each of them has a probability beta to go out of the seed bank. Right? So that means that your whole tree and the genealogy, the size of the genealogy is affected, is rescaled. The mutation, as I mentioned, appears only in plants which are above ground. Okay, we assume seeds do not mutate. It's also an assumption one can discuss, but from the literature in plant, there is no evidence that much mutation should happen in seeds. So that's why we took this assumption. And that means that the mutation rate, the scale mutation rate, is also multiplied by beta, because you do less mutation because you do mutation only when you go out of the seed bank. What we did as assumptions, which are totally unproven, so also if someone wants to really do that rigorously, I'm really open to discuss about this. We assume that the recombination rate and migration rate, as they also affect only 1 lineages, are also scaled by beta. Right? So it's the same principle that recombination and migration only happens also above ground. Okay, so when we did that, we wanted to do a certain number of analyses. So the first one is we wanted to show, can we infer or do some inference on this parameter based on genetic data? Then the second is, what are the consequences of the seed bank if we have different demographic events in the past? So for example, change in population size. And the third one is, what happens if we scale it up to some speciation model? So if we look at divergence between different species which date longer time ago, does the seed bank also affect our inference in these cases? Okay, so I show very quickly some of these results because these are a bit older already, but just to set up what are the questions. So the first result, we wanted to show to infer the parameter of the seed bank. The main problem, of course, is nothing is simple, is that the species also are spatially structured, so we have many populations which are connected by migration. So that means we also need to take into account the spatial structuring of populations. So we use some of the models by John Wakely of metapopulation and some of the scaling which are in these models. And so that means that the effective population size or population size depends on the number of individuals per population, which is still this N census, the number of demes, the number of populations, and the migration rate between them. Right? And then in addition, we plug in our seed bank model where each population can have a seed bank with a germination rate B. Okay? So then we did a lot of so we use all this by simulations, of course, because it gets a very multidimensional problem. We have different sequence data and so on. I'll skip the details. What at the end we could come up with, and we were really happy, is we could estimate, have some Brusel density for the germination rate B for these two species. So these two species have different ecology, but doesn't matter really here. Okay, and so where the power lies in this method and what is very crucial is that actually the estimate of B is possible only because we have some prior on the census size. Right? So that means that we have some ideas here of these numbers. So we can put some prior of between, let's say, 10 and 1,000. Right? If you do not have any prior information on this, there is no way that you can estimate any of this parameter, because in the model, by definition, the beta is a function of B and M, and if you know nothing about it, you cannot estimate any of them. Right? So this is one of the biggest limitation. So in that system, we have some information, but that means not very generally applicable to any system necessarily. So what we wanted to look as a second outcome of that study is also what's happening. So as I mentioned, the tree is rescaled by this factor 1 over beta square. And so what we wondered is if there is demography happening in the past, so I'm sorry, for biologists, the time is running that way towards the present. So if there was, for example, change in population size over time, if the change occurs at a given point in time, if we change the genealogy size, that means that potentially the number of coalescent changes, or it will be changed along the tree, or the tree will be rescaled. That means that we might not have the same information depending on the size of the tree. So we wanted to look at this with Daniel Zivkovic, and so what he did is to reuse some of the results by Griffiths and Tavaré, where we basically use the same process to calculate the probability of the ancestral process to have G lineages. I think the equations don't look really good, but okay, you can look at them later if you want. The main point is basically we get exactly the same results that they had, and we have, so the classical here, choose k in 2, which is the usual Kingman coalescent. We scale it by our factor beta square again, and this function here, this integral of rho s, is basically the function of the change in population size which happens over time. Okay? So you can just rescale the part of the tree depending on the change in population size piecewise. Okay, so this. Yeah, I just mentioned it just now. Yeah, so I'll just maybe explain and so of course we can calculate also from this the expected time of each of the coalescent events. And then what we can calculate out of this is the side frequency spectrum, right? And so this is here the relative side frequency spectrum that we can get for any sample size n with any change in population size that we want, assuming a value of beta. Okay? And so what we did here is just some simple calculation that if you have, so here different demography, so this is time from present to the past, and you have here different 3 types of demography, so there's 3 lines. Okay, so you can see there is a very recent expansion forward in time, a bit less recent and quite old, and this demography actually corresponds to different simulation with different beta value. Okay, so here beta 1 means there is no seed bank, and the usual coalescent everybody is using, and these are all the seed bank with beta 0.2. And when we do that, we can generate the side frequency spectrum you can see here. So these are the 3 colors in black, gray, and white, and you can see they are exactly identical. I think we choose them on purpose, right? So the idea is that now the problem is that means that different past demography can result in the same side frequency spectrum or allele frequency spectrum, which of course is a bit of a problem if you want to do an inference, right?
So do you have the same mutation rate before rescaling?
Yes,
Because
yes,
the
yes.
shape of the tree is the same, but the branch length will be...
So here this is the relative side frequency spectrum, right? So that means that when there is the seed bank, actually the relative side frequency spectrum does not change, is not affected, because it's a Kingman coalescent just rescaled. However, the number of segregating sites is of course higher when there is a seed bank, because the tree is scaled by beta square, the mutation rate is scaled by beta, so that means we still have 1 over beta more diversity due to the seed bank.
So it's just saying that the relative fraction of singletons...
Yep, exactly. So if we would take the total number, they are different, right? The total number of sites. Yeah. The problem is basically, to go back to what I mentioned from the first result, is that if you have no way to know the end census in the population, so the census size, you cannot know if your higher number of segregating sites is due to a higher population size or to a longer seed bank. That's the problem that we circle around. Okay, so just to make things even worse, we took a more complex demography with a typical bottleneck, which happens in many species. And then what we could do is, again, to do the simulation for, or the calculation of the side frequency spectrum for, again, the 3 types of seed banks, so no seed bank and 2 very old. And what is interesting, so I don't want to describe the side frequency spectrum in detail, but just to, in a nutshell, what's happening is that if you have no seed bank, you have a lot of information about the recent events, because of course most of the coalescent event happens in this phase, where if you have an intermediate seed bank, which is, let's say, intermediate long, you get a lot of information about this bottleneck phase, and if the seed bank is very long, you get information about this phase. So the side frequency spectrum changes accordingly, that we recover information about different time of the past if we have different seed banks. So we would just expect that because we scale the tree differently. So the good news, in a way, for biologists, it means that for inference, the seed bank gave access to more ancient demography. So that's quite nice, potentially, that we cannot recover with a normal Kingman scale coalescent. However, the bad news is that you need to know this beta, because if you don't know it, you cannot know anything about your demography. So again, we circle around that problem. So the third, I think at that time we should have stopped and stopped doing research on this, but of course I was a bit greedy, so I thought, okay, let's extend this idea. And now we wanted to look at speciation. So if we have now a model again of one ancestral population splitting into 2, so this can be 2 incipient species, for example. And what we wanted to look is at the speciation between these 2 solanum species, so these 2 tomato species, because they recently diverged. So that means you still find some polymorphism which is shared between the species. Right? And so what we wanted to know is this shared polymorphism due to introgression, so selected integration from one species to the next, or is it just due to the remaining of the ancestral polymorphism still segregating between the 2? The idea behind that at that time, as you will see, the results do not look very good, but our main idea was to try to look at balancing selection, so genes which maintain several alleles in each of the species due to selection, and to look if some of these alleles seem to migrate from one species to the next, so if there will be selection on them. Okay, so what we did, we started doing some sequencing. We looked at different genes, which were our candidates. We had different reference genes. We have quite a few SNP because, as I mentioned, the species are very polymorphic, so it's quite nice. We have a lot of samples of different plant population from Peru and Chile, and so on. So we do a lot of work in the lab, clean up the data, and so on. And we built our little model with an ancestral population size to derive populations which can exchange migrants and a time since speciation tau. And what we do is that we use here, so there are likelihood method to do that. However, we didn't feel confident enough to change to put our seed bank model in there also, so we used our own built approximate Bayesian computation method to do that. For some simple models, the results are very similar, so believe me, I can show you some more examples. And so what we did is also to use the so-called joint side frequency spectrum. So this is a matrix which summarizes the information of the SNP frequency between population 1 and population 2. So you do a matrix and you look which SNP in which frequency it is. And this was proposed by John Wakely in 97. And so we use this as a frequency or as a statistic of our SNPs. And then we run our ABC. We did many controls to check different parameters. these are how the posterior look like. You don't need to look at what are the parameters. The main interesting one is, of course, the time of divergence tau. And what we recovered was something on the order of 1 million years ago. It's slightly overestimating the previous results which were found by Dirk also, but our confidence interval encompasses what has been shown before. So we were pretty happy that our Bayesian method with seed banks, without seed bank, gives an accurate estimate based on roughly the same data as other people. Of course, the main problem runs with the seed bank that we do the same, and as we didn't have much prior for much information about what's happening in the world species level, we didn't put much prior, which was probably a wrong idea, because then we get a time of divergence which is 10 to the power 9, and if you don't have any idea what that means, this is the history of the planet. That means this tomato would have speciated before even the plants were even appeared, right? So that's why you should always stop at some point doing inference, right? So this is, I think, if you think about it, that's a pretty obvious result because you scale the tree by this factor beta square. But we still thought, well, somehow maybe in this model, there will be the fact that the shared allele and so on are so high, this can be that maybe we will still estimate something which is low divergence time enough. So what this means at the end is that basically there are two problems I think out of this. That means that the seed bank can really keep potentially shared allele, which are ancestral polymorphism for a long time. That's because the lineage escape drift and they stay in the seed bank. And the second one, which I think is the main problem, is probably that seed bank adaptation might change over time. So this parameter beta actually probably is a function of time. So it might evolve depending on the environment and so on. And if you start doing this, of course, the tree is not scaled uniformly by beta square, but it will scale by different value over time. So how do we now approach some of these problems? So first, I will mention some new data that we gathered, a very quick overview that will help us in going in some direction. And then I'll show some little modeling attempt afterwards. So now what we wanted to do to go understand a bit better our populations, we focus only on this one species, because it's already complicated enough. And we have all these populations where we have seeds and plants which were collected by colleagues in the US, in UC Davis. So we can get the seeds and we can also grow them in the glasshouse and then do some sequencing, for example. So we have all these plants which come from different environments. You can see some very dry, some less dry, depending on their place. Just to emphasize maybe for curiosity, so those populations, for example, are found at above 3,000 meters of altitude, so they can be really cold and snowy and so on in the winter, whereas those ones are really on the coast where, for example, it's really dry and very salty environment. So they are very different adaptations, potentially. They are the same species, you can cross them, right? So what we wanted to do, we sequence, or we take this population, we took 25 plants per population, but at that time, it was still a couple of years ago, sequencing was not so cheap. So we did only a limited amount of genes, but as we have many SNPs for inference, we don't think that's really a big problem. And also, I didn't mention, but we have also a high recombination rate in this species, so in general, the SNPs are more or less independent, so we can assume that the linkage disequilibrium is very limited. So we choose some candidate genes which are related to some adaptation we wanted to look at. I'll not mention as part of this story. And when we look at the data, so we did many analysis. I'll just summarize here the main point. This is here in the x-axis, the latitude, so from north to south. Each dot is a population, okay, so the average over all the genes. And what we see, so this is the theta Watterson and pi. So pi is the pairwise comparison between individuals. And so you can see clearly that the diversity decreases from north to south. So the black dot are for all the genes, and this little diamond, little triangle, are from the reference genes. So they are very similar in most of the time. So this indicates, so we see a decrease of variation. We see that we look at pi A, pi S. So pi A is the same idea, so you take the pi. But now we can separate the sites which are synonymous and non-synonymous in our coding region. The rationale is just that the non-synonymous sites are expected to be on the stronger selection, either purifying or positive, than the synonymous sites. Right? So we see an increase from north to south, though it's more noisy, as you can already see. And then what we look here is the TGMSD, which reflects the skew in the site frequency spectrum. Right? So negative TGMSD is more low frequency variant and high frequency variants. Higher TGMSD, positive TGMSD means more intermediate frequency variants. So all in all, when we look at all this data, what we concluded, and we did other analysis, so we have different genetic groups, so populations which seem more closely related than to each other than others. And we have potentially a north to south colonization, so probably the origin of the species is in the north, and then there was colonization along two axes, so one along the coast with some specific adaptation, and another one in a high altitude with all the specific adaptation. Okay. Believe me, I don't show you all the results, but we did. And I just want to cut the story to the main point. So we did also some other method to detect selection and so on, and what we find is that we have stronger purifying selection in the northern population, but we find traces of local adaptation in this kind of peripheral groups on the coast or in a high altitude. So what does it have to do now with all of this with the seed bank? The question is, how do we interpret all these results? And one way to interpret is there was a very simple colonization from north to south, and along the colonization there was small population sizes because of course you colonize new habitats, so it's possible you get bottlenecks and so on along the way. And with adaptation. The problem with that scenario and what many people would expect is that you should find very low population sizes here, because this population still, if there was really bottleneck, you should have strong adaptation and strong genetic drift. When we look at this data, you still find that the population sizes are very large, so the genetic diversity is still very large compared to many other species. So an alternative hypothesis, I'm willing to also discuss other ones, but an alternative hypothesis that we propose is that maybe there is a change in the seed bank between these different populations. So there might have been indeed, it is not mutually exclusive, a north to south colonization. However, we could maybe think that there was a change in the seed bank, and what made us thought about this is that we look at the seed weight. So this is here a graph, again in function of the latitude from north to south. And this is here the mean seed weight. So this is basically an average of the seed size for each population. And what we see is that there is, and we have more data to confirm that now, that you see an increase that the seed in the north seems to be smaller than the seeds in the south of the range. So why this is important? Because the seed size is very key for seed banks. So we know that the small seeds tend to stay in the soil for longer because they can get buried deeper, and then it takes longer until they come up again. And so this in ecology has been demonstrated many times that this will be a key determinant of the seed bank length. So of course our hypothesis now, what we would like to test is whether the initial state of the species was with small seeds and very long seed bank, and maybe by colonizing new habitat there was a change in the seed bank adaptation so that the seed bank gets basically shorter and shorter. Right? Still, to some extent, that we maintain still a lot of diversity. Now, if you think about the model I presented before, that means we have two ways. Either we change B, so the germination rate itself has changed, has evolved, or we decrease M, right? Because we can also reduce the maximum amount of time seeds spend in a seed bank. Now, we have no way of testing this, so that's a nice hypothesis. So if you have any idea, welcome to hear it. So I'll show now some little models that we try to build, maybe with the aim of at some point testing this hypothesis. I think I just want to emphasize the interesting thing about this hypothesis is also that that would mean that changing this ecological adaptation of seed bank would kind of feedback on the genetic diversity that we observe in a genome, and that of course feedback, for example, on drift and the strength of selection that you get then in the populations. So ecological adaptation might be fast enough to then completely change the rate of adaptation in a genome, if that works. Okay, so I will now quickly show you some very much work in progress. So I don't claim that any of these results is really true and well demonstrated. So don't kill me. And basically the idea is we want to get rid about that problem, or I would like to get rid of that problem that we need to know so much information about the ecology to estimate any of these parameters. So the first idea is to think now, okay, we have taken this nice model where we know we take n to the limit and we get the Kingman coalescent and n tends to infinity. Of course, in reality, we know it's not really true. And with the idea of some of the paper by Takahashi and Wakely, where they showed this oversampling effect, so if you sample your sample size little n on the order of magnitude of big N, your population size, then you get all this multiple or simultaneous merger in the first generations. And the idea is to maybe look if we could do that also in our model of seed bank. So the idea is very simple. We do a forward simulator of the Chi model I showed you before. And then we have here a small sample size 20. And then for convenience of the simulation, we keep m times n census to 3 different values, so 500, 1,000, or 3,000. So that means I keep constant all the plants which are available, right, over the m generations. And I will just change now m. So that means that I can have more or less plant per generation. And when we did that, we didn't expect much to come out of it. So this is here the graph. So this is the m value, so no seed bank, and then to higher m, so longer seed bank. And this is here the total number of multiple and simultaneous merger that we could observe in our simulations. I have to note that here we didn't scale it properly, so that's not very nice, but we did 200 simulation, and 200 means here that this dot has on average 1 multiple merger, right? But we can scale the parameter differently to get more. Just an overview. And so the main idea, I hope to convince you that when you look in blue, so that means nm is 500, in red it's 1,000, and in green 3,000. I hope to convince you that you see that the curve and the plateau is different. So that means that we have maybe a chance when we look at, for example, small m's or short-lived seed bank, there is maybe a big difference already when m is 4 or 6 or 10. We might be able to see that in this number of multiple and simultaneous merger. Right. Here, this is a simulation for a uniform BI, so every BI is 1 over m. We did it also with a geometric. B is 0.1. It's slightly different, the shape, if you compare the two. I will not put too much emphasis on small differences and jump, because we had problem with discrete problems in the simulation, so that's why you have this little jump. But we would expect that we should get a smooth distribution. And so I think these encouraging results mean that potentially, at least for small m, so for big m, it seems very difficult to disentangle because you see it's very flat, the line. But in this part here, for small m, it might be possible to disentangle the effect of m and n census. So that means that increasing m or n does not have the same effect on the rate of coalescence in this finite setting that you can allow for multiple merger to happen. So of course, I think what this means also is that if we would increase our sample size, we would expect to even generate more of this multiple simultaneous merger, and that means we should maybe be able to detect that in the side frequency spectrum, for example. I think typically one of the problem, again, is that for our species, which has very big population size, potentially we would need sample size on the order of probably 1,000 or more. So, okay, that's one of the downside. Okay, and so I think in the last 5 minutes, right, I will just allude very quickly to probably what most of you would be interested in, but I think I cannot explain every detail properly because, as I mentioned, this is a collaboration with Johannes Müller, so we did this together. I'm really learning a lot about how to do demonstration and proof, so I really, it's very valuable for me. But I think I cannot explain all the details of this derivation. So this is work in progress, and the rigorous proof is not finished. So if you have any idea or you want to discuss about this, I'll be happy to. So what we wanted to do is to do a forward version, a diffusion model, which basically mimics the Chi model. Right? So instead of having it backward with a coalescent, we want to do it just forward. So what we did is we do a population of n plants. We have 2 genotypes, A and B, so it's very classical, as in the Moran model. So we take example of the Moran model and just extend it here with a seed bank compartment. So we have some age of the seeds, which are distributed with some age function. And then we have death and rebirth of a plant, creation of seeds, and then death of the seeds. So how do we do that? So we have death of plants, so the plant die at rate mu. Okay? And of course, they will have this ability of germination, which is the sum of the BA here, which defines the age structure of the seed bank. I think for the proof, we need that BA is smooth and has a bounded support, and I will explain later how we made it in the next slide. And what we also do is that we assume that the seed bank in that case is completely deterministic, so there is no stochasticity happening in the seed bank. So in other words, that means that we do not remove the seed. So we kill a plant, it is replaced by a seed. So the population size remains constant always, but the seed that we remove from the bank is not gone. So we assume that the plant have produced enough seeds of one type so that there are always some which are in the seed bank. Of course, assuming that they are not dead, because we have a bounded time for which they can stay in the seed bank. Then there is production of seeds, so the plant produces seeds at rate beta, and then you can define that the A seed will give seeds at the rate beta xt, and beta n minus xt for the type B seeds. And we have death of seeds. Actually, this mu s, we don't really need it, because then afterwards we plugged it directly into the definition of the age structure that we can bound. Okay, this is the point where I will start skipping things for sake of time, and also because I think it will take much more time to explain and to remember how we did it, so and what I understood. So forgive me, and if you want to look at it in detail, I'll be happy to discuss. What we can calculate is now the probability for plant dies that it is replaced by a type A plant. So we do this for interval t tau. And so we can integrate to this B tilde here, which is basically the age structure of all the plants of type A which can germinate. And then we can scale this by the total number of plants of the old age structure of type A and B. And then we can find the probability for type A, which is given by this. And then we can start building our model where we can calculate the probability that we have either one more or one less type A plant. And I think that's exactly like in the Moran model. That's the usual writing. And at the end, after many steps, we managed, I think we were quite happy to prove that we have SDDE, so that's the stochastic delay differential equation, basically. And we scale here with eta is, epsilon, sorry, is 1 over n. And then we can use this, so xt, sorry, is the frequency of allele A, right? So basically xt over n. And so then we can, well, if you're familiar, I think that's exactly the same writing as the Moran model, except we have a couple of coefficients which appear. And I think the whole bulk of the proof at the end is to now simplify life. First, we do a uniform distribution for this interval 0 to big A. Actually, this big A would be my m in the chi model. And so we define this BA function. And in that case, we can rewrite the equation, so this just change of notations. And we were happy to show that the time scale that we can apply to rescale this to a Moran model is this function here, so 1 plus a over 2 to the power minus 1. And how this happens, and I think there we still have to work the proof in detail, so I'll discuss with Jochen already some ways of doing it rigorously. But in the limit of n tend to infinity, that means epsilon tend to 0, we can basically, the drift term is vanishing, or at least is on the order of the noise that we can then get rid of. And then, of course, we wanted to do a more general, so that's very then easier to just now do any general BA function, which will be basically this phi A, and phi A defines the age structure of the seeds in the seed bank, and we find out that the rescaling that we can apply is this factor 1 plus e to b. We just wrote it because it's the same as in the chi paper, so you can exactly make the matching. And this E of B is basically, of course, the expected time that seeds spend in the seed bank. And in contrast to what Jochen will show, this is where we were really happy that we can get a rescaling to correct to a Moran model, and that means that we really have exactly the same model as in the chi paper. And as we were, again, greedy a little bit, it's just 1 minute, we basically extended for weak selection. And so that means we define this selection coefficient, S and SP. What they mean is basically you can have selection on how many seeds you produce, or how long the seeds can remain in the seed bank. So some can stay longer than another type. And if we do that, and we assign this to 1 plus sigma over n, in the usual notation, then we can again show that the drift and the diffusion coefficient are again scaled by the same parameter, which would mean that selection is indeed slower if you follow this model. So that, as far as I'm aware, is the first time that it somehow proved that this intuition is really true. And with this, I will thank you for your attention. And these are the people in my group who are currently trying to do some work on tomato and modeling to go further in this direction. Thank you.
So because we are being taped, I've been asked to hand the microphone to people who are asking questions. So do we have some questions for Aurélien? Yes, sir.
Yeah, so my question is, when you go backward in time in the Cas-Kronen-Lascaux model, you get a time change of the Killman coalescent. But forward in time, one would expect a Greif-Fisher diffusion. I don't know if I didn't see it in the notation, but is that a Greif-Fisher diffusion?
So what I showed was for the Moran model, basically. So you just have the time scale change to rescale to the Moran model. We also did it for discrete time for the Wright-Fisher, and we also get the equivalent.
So it is a Wright-Fisher diffusion, slowed down by the factor b squared. Okay.
So you were suggesting using the multiple merger frequency to get at the sensor size, but presumably you could get a much more direct estimate by looking at information from recombination and LD over a short number of generations. I mean, in principle, you can construct relationships back a few generations.
Yeah, so that's exactly, I cannot mention everything. That's also what we are also doing. I think the idea of using recombination, I think, will be probably very informative, because as we have this different time scale of the coalescent scale is scaled by beta square, but the recombination, if it's true, is scaled by beta only. That means that if we go along the chromosome, our aim is to build a kind of a hidden Markov model where we can go along the chromosome and estimate the difference between the time to the most recent and common ancestor, which is scaled in beta square, and the recombination rate, which will be scaled in beta. So that means that we might have a way of estimating beta directly from this going along the chromosome. Yeah, but yeah, we're still working on developing that, so I cannot say yet if it works. But
perhaps one more question, if we. Okay, so if not, let's thank.
Thank you.